Good morning, children. In the previous class, already we have discussed about the process of photosynthesis, definition for photosynthesis, equations for photosynthesis, and also we have discussed that starch is the index for photosynthesis. Today, we are going to discuss the materials essential for the process of photosynthesis. So, what are the materials that are essential for photosynthesis? Already from the equation of the photosynthesis, we came to know that carbon dioxide, water, sunlight, and chloroplast are essential for photosynthesis. Today, we will discuss about the water and photosynthesis. Water is essential for photosynthesis. Children, in your 7th class, already we have studied that Von Helmont experiment. The Von Helmont experiment, from that already we came to know that water was important for the plant growth as well as it helps in increasing the mass of the plant or weight of the plant or body mass of the plant. By that time, photosynthesis was unknown. How the body mass of the plant increases, by that time it was not revealed. But during that time, Von Helmont, he come to the conclusion that water is essential for the growth of the plant and water is also responsible for the increase in the body mass of the plant. Okay, children? Next. So from that already we came to know that water is essential for photosynthesis. Already know through the roots of the plants they absorb water and minerals which is supplied to the leaves, which are the food factory of the plants where the actual photosynthesis will take place. So water is essential for photosynthesis. Actually, in fact, water is not only essential for photosynthesis, water is also essential for each and every metabolic activity present in our body. So most of the metabolic activities they require water, so as photosynthesis also require water. Next we will discuss air and photosynthesis. So actually scientists for about 300 years they took to understand that water is essential for photosynthesis. Carbon dioxide gas is also essential for photosynthesis. Sunlight is essential for photosynthesis, and chloroplast and chlorophyll pigments are essential for photosynthesis. And there are many other things still they are yet to discover. So what since last 300 years different scientists they have understood regarding the process of photosynthesis. Just we are explaining it. Okay, children, already know in previous classes also in 7th class also you have heard about Joseph Priestley. Joseph Priestley, he has conducted Belger experiment during 1770s to 1780s. And in 1774, he discovered that oxygen gas is present in the air. Understand, children? And oxygen gas is essential for our breathing, already know. And oxygen is also very much helpful for the burning of the candle. Okay, children? So oxygen is essential for respiration and oxygen is also helpful in combustion process. So oxygen, who discovered oxygen? Oxygen was discovered by Joseph Priestley. In the year 1774, he discovered the oxygen gas. And later, in another year, Lavoisier, he has named that gas as oxygen. So oxygen was discovered by Joseph Priestley and it was named as oxygen by Lavoisier. Okay, children? Now let us see how Priestley come to understand that oxygen is very essential for respiration and oxygen is also essential for combustion and how photosynthesis releases oxygen. Okay, children. So first of all, Joseph Priestley, he has taken a bell jar, and in that bell jar he has burned a candle. He has burned a candle and he has covered the candle with a bell jar. After some time, he found that the candle has put off with a pop sound. That means, as long as the gas that is required for combustion was there in the bell jar, inside the bell jar, air is there, and in that air there is some component which is essential for the burning of the candle. As soon as the gas present inside the bell jar was exhausted, the candle was put off. Similarly, he has taken a rat. Rat is an animal, living organism. That is an animal. And he has kept a rat and he has covered the rat with a bell jar. After some time, the rat felt suffocation. And after some time, the rat also died because of asphyxiation. Because of a process called asphyxiation, oxygen on the gas. Okay, the rat has not received the gas. As long as the gas present in the air inside the bell jar was present, the rat was moving. It was alive. But slowly, when the gas that is required for breathing is exhausted, the rat started suffocation and died. So from this, Joseph Priestley has concluded that for burning of the candle and for breathing of the rat, there is some component in the air that is essential. Okay, children. Later, he has done another experiment. He has burned the candle. He has taken another rat, and he has kept a mint plant, and he has covered all the three with a bell jar. He observed that the candle is continuing to burn, the rat also moving without feeling suffocation. So from this he has concluded that the gas that is present in the air that is required for burning of the candle and for breathing of the rat are actually produced by the mint plant. That is the reason why in the presence of the mint plant, the candle continued to burn and the rat also continued to breathe and became alive. From this, he has concluded that there is a component in the gas, or there is a gas in the air, or a component in the air that is required for breathing in animals and burning for candle, and it is produced by the green plants. So he has discovered that gas, but he has not named it. So he has named that gas by Lavoisier. The oxygen gas was named by Lavoisier and was discovered by Joseph Priestley. Okay, children. So we came to conclusion that both water and air are essential for photosynthesis. And children, can you tell me how carbon dioxide and water are exchanged in the leaves? Carbon dioxide from the atmosphere can enter into the leaf for photosynthesis. Okay, through a structure called a stomata. A stomata will have two guard cells, and there is a small opening that is called a stoma. So two guard cells and an opening called a stoma. So through the stoma, the carbon dioxide gas enters into the leaves, and the oxygen that is produced goes out from the leaves. So the exchange of gases in the leaves takes place through stomata. Okay? and sometimes in the stems also there are processes called as lenticels. So stomata in the leaves and lenticels in the stems, they are responsible for exchange of gases between the surrounding and between the plant. Okay, children. Now we will discuss that an experiment to prove that carbon dioxide is essential for photosynthesis. For children, carbon dioxide is essential for photosynthesis. For any experiment related to photosynthesis, first of all, we have to discuss the aim of the experiment. Next, apparatus required for the experiment. Next, equation, photosynthesis, equation you have to write. Next, we will discuss about the procedure to conduct the experiment. After conducting the experiment, we will observe, there is observation. What is our observation from that experiment? Next, we will come to a conclusion, that is called inference, or you can write result. So what is the aim of your experiment? In the aim, what is to be proved? Here it will be proved. In the result, you must have to prove the aim of your experiment. And if necessary, you have to take some precautions. So, in this experiment to prove that carbon dioxide is essential for photosynthesis, our aim is to prove that carbon dioxide is essential for photosynthesis. Okay, children, for this experiment, we need to take some apparatus. You can see some of the apparatus as mentioned in the picture. Okay, so we have taken a potted plant. Then a wide mouthed bottle which is covered with a split cork, and inside the bottle 5 to 10 milliliter of potassium hydroxide solution is taken. Potassium hydroxide solution. Okay, children? And finally, we have to test the leaf for the presence of starch by conducting iodine experiment. Already in the previous class, we have done an experiment to prove that starch is the index for photosynthesis. So already I told you, if photosynthesis has occurred in a plant, starch must be formed, and we can test the presence of starch by conducting iodine test. Okay, children? So now let us discuss about the procedure. First of all, for conducting any photosynthetic experiment, first of all we have to destarch the potted plant. What do you mean by destarching? Already you know, children, once photosynthesis takes place by the green plants, glucose is synthesized, which is converted into starch, and starch is stored in the leaves. Okay, children? And it is stored as a reserve food also. Understand? So if already starch is present in the leaves and we are conducting whether photosynthesis has occurred or not experiment, then it may interfere with our result. That's why we must have to destarch the potted plant by keeping the potted plant in dark for about a week or at least 3 to 4 days. So in that 3 to 4 days, photosynthesis will not take place in the darkness. Already you know, whatever the reserve food is stored, starch is stored in the leaves, that will be consumed by the plant itself. So the leaf will be free from starch. The leaf will be free from the starch. Understand, children? So to keep the leaves discharged, the potted plant has to be kept in the darkness for about 3 to 4 days. If possible, 1 week also you can put, no problem, nothing will happen. The plant will not die. Okay, children? On the day of experiment, we have already selected a healthy potted plant and we have kept in the darkness for about 3 to 4 days. On the day of experiment, we will select a healthy leaf, a long and a narrow leaf we will select. From the potted plant, we will select a long and narrow leaf. Understand, children? We will take a transparent wide mouth bottle. A transparent, usually the bottle is made up of glass, so glass is transparent. So you will take a glass bottle which will have a wide mouth. That is called wide mouth bottle. And you will take a cork and you will put a split in the cork. Okay, children? And you will remove the cork. You will add 5 to 10 milliliters of potassium hydroxide solution in it. You will take 5 to 10 milliliters of potassium hydroxide solution in the wide mouth bottle. You will take a split cork and you will insert the long narrow leaf in such a way that half of the leaf will be outside the cork and half of the leaf will be inside the bottle. You will close the cork. You will fit the cork into the wide mouth bottle. Half of the leaf will be there inside the bottle and half of the leaf will be there outside the bottle. Okay, children? Now we have already poured water to the potted plants, and we will take the entire setup and you will keep it in the sunlight. So why you are keeping the entire setup in the sunlight? Photosynthesis will take place. Okay, children? Now after keeping the entire setup for about 4 to 5 hours, we will remove the leaf from the potted plant and we will take out the leaf from the wide mouth bottle and we will test the presence of starch by conducting iodine test. Okay, children? Now you will see. Now what is our observation? Next point is our observation. What we will observe? We will observe that the part of the leaf which is outside the bottle has turned into black blue color. Whereas the part of the leaf which is inside the bottle has no change in color. If there is no change in color, what does it mean? This means photosynthesis doesn't take place inside the bottle, but photosynthesis will take place outside the bottle. Okay, children? Now, inference or the result. So, from this experiment, we will conclude that outside the bottle, all the factors that are essential for photosynthesis are present. Sunlight is present, water is present, because what is absorbed through the roots. In the leaves already water is there. In the leaves already chloroplast is also present. Carbon dioxide is also present in the air. But inside the bottle, all the factors that are essential for photosynthesis are present except carbon dioxide. Because we have taken potassium hydroxide solution, and potassium hydroxide solution will absorb the carbon dioxide that are present inside the air in the bottle. So inside the bottle, wide mouth bottle, all the factors essential for photosynthesis are present except carbon dioxide. And because there is no carbon dioxide inside the bottle, so the leaf which is present inside the bottle has not turned into black blue color, which is the index for photosynthesis. Starch is not present in the part of the leaf that is present inside the bottle. Because starch is not present and starch is the index of photosynthesis, so photosynthesis also doesn't take place. Did you understand, children? Here in this experiment, we have to take a precaution. Last one, precaution. The leaf inside the bottle should not touch to the potassium hydroxide solution. Understand, children? Should not touch to the potassium hydroxide solution, because already potassium hydroxide solution has absorbed the carbon dioxide. If the leaf will touch the potassium hydroxide solution, carbon dioxide may enter into the leaf and photosynthesis may take place. So to avoid the interference we will not allow the leaf inside the bottle to touch the potassium hydroxide solution. So from this experiment we can say that carbon dioxide is essential for photosynthesis. Understand, children? Thank you very much.